Thank you very much, and thanks for the joyful day, talking to many of you guys here in the audience. So it's time for you guys to listen to me, so hopefully it will be as interesting. Please stop me at any time, actually, if you have a question or you want to criticize something or discuss something, then feel free, of course, right? So today I'm actually going to talk about work in my lab that actually is another pillar, which is really our interest to understand genophenotype relationships. I don't know if this is well attached or not. It looks good? Okay, good. And I always start with this slide because I think it brings home the message really well, in a sense that we're all a bit envious about this lady who at her 100th birthday basically is happily smoking a cigarette, and then you kind of have to wonder whether basically this lady is incredibly lucky with the genes she inherited, and so she's a very robust person to all kinds of environmental insults, or whether she's just a lucky person in general and somehow basically defied her own genetics. So going from genotype to phenotype is something that we like, many of you do as well. And I think one of our interests is not only actually to link those two, but to really try to understand the underlying mechanisms through, for example, the application of different kinds of molecular techniques such as gene expression analysis, chromatin state analysis, and try to understand how these molecular layers interact, so to speak, in order to actually produce a certain phenotype. Okay. So very early in the lab, when we started it, we realized that actually this is darn difficult to study in humans for many reasons. And so we really actually adore the kind of standardization ability that model organisms provide, and none the better, of course, than Drosophila, which many of you, of course, know very well and like as much. And so that's why early on, actually, in the process, we got involved in Trudy McKay's effort to really generate a new resource. And now the slide is holding me back a bit. Let me see if I can move it forward as such. Yes, I can. And so I think I saw that Trudy was here maybe not too long ago, so most of you should be familiar with the Drosophila Genetic Reference Panel. Just for those who are not, this consists of about 205 lines. I mean, I think Trudy started originally with more than 1,000 flies from a local fruit market, through then local inbreeding or sister-brother mating derived many hundreds of lines, but only basically 205 really survived this stringent inbreeding process. And of those, I would say probably only 140 are truly robust to work with. Nonetheless, once you establish this, of course, then you can use them very easily for phenotyping. You can also look at, of course, the dynamics of genetic variation, and then you can also apply all kinds of molecular techniques. And so, as indicated very early on, Andreas Mazura is then a PhD student in my lab. He now actually started his own company, and some of the technologies he developed in the lab paired up with the postdoc in Trudy's lab to actually map genomic variation in the DGRP lines. And so this is the result of all of that, actually summarized in one slide, where we, of course, saw tremendous variation going from, of course, a lot of SNPs all the way to small and insertion and deletions, and then, of course, larger structural variation. So the numbers are actually indicated here. A great resource to work with, because on average, basically, every 10 nucleotides, you roughly have a variant. So this is a highly polymorphic population that is bound to also show a lot of phenotypic diversity. But this kind of illustrates also one of the problems, which is basically that this is only dealing with nuclear genomic variation, which I think we all agree, of course, is probably the most important determinator of certain phenotypic variation that we see in this population. Nonetheless, we also know that actually another genome, namely that of the mitochondria, is also increasingly linked actually to certain phenotypes. For example, certain diseases such as obesity, which we study in the lab, type 2 diabetes, neurodegenerative diseases, and schizophrenia, and so forth, right? So we kind of wondered, should we also actually consider the mitochondrial genome and its variation in this population? And if so, basically, how has this already been addressed? Has this been addressed comprehensively? And how does this relate, for example, to phenotypic variation in the DGRP population itself? And interestingly enough, there are other model organism resources out there, but the DGRP lines are somewhat unique in that they're genetically independent, and so each line basically has its own mitochondrial DNA haplotype, so to speak. But so far, actually, in our own studies, which we just used whole organism genome reads, as well as a study actually published a little bit earlier, from Richardson and Casey Bergman were involved in this. There was already an attempt to sort of map mitochondrial DNA variation, but I think, again, not to the most comprehensive fashion, because it was also not really the focus of the respective papers. So we decided to address this. We is actually Roel and Masha. Roel recently graduated from the lab. Masha is still in the lab. So Roel did all the experimental work, and Masha most of the bioinformatic analyses. And so in order to address this particular problem, Roel devised actually a new methodology to really derive highly enriched mitochondrial DNA from Drosophila flies, which is indicated here. So you isolate for mitochondria, then you perform DNA extraction, and then you actually tagment with TN5, such that you can now actually sequence in a multiplexed fashion actually many of the lines together and still actually reach high coverage for each of the mitochondrial DNA lines. And if my slide will move forward, again, it's sort of stuck, not sure why, but this is kind of a summary of what we discovered. So in total, basically, we detected 263 variants actually in the mitochondrial genome. Just to say that we actually sort of disregarded this AT-rich repeat region, which is very hard to resolve, and we had an interesting discussion this afternoon on perhaps how we could improve this actually on using better assembly methods. So please disregard this for now. We're not considering this here. But so the rest of the mitochondrial genome is actually still containing quite a lot of variants, 241, 15 indels, which affect those mitochondrial genes and also multinucleotide polymorphisms. Now, I have to say, of course, that most of these variants actually occur at low allele frequency. This probably has a reason. I mean, this is, of course, a very gene-dense genome. And so probably, many variants that actually might affect in a deleterious fashion actually certain mitochondrial genes might be quickly purified, probably explaining why most of them are actually low allele frequency. Okay. So this actually is, to our knowledge now, the most comprehensive variant catalog within the DGRP for mitochondrial DNA variation. It added actually a sort of 30 or 40 variants beyond the ones that had already mapped before. So it made it actually an interesting resource now to actually further categorize this and see how this variation now links to phenotypic variation. Okay. Now, as you may have heard from Trudy also, one of the peculiar things actually of the DGRP is that actually very little population structure has actually been detected based on the nuclear genomic variation. This is somewhat illustrated here, where you actually see the pairwise relationship between individual lines. So you can have a total of 20,000 or so pairwise comparisons. And you can see that actually in terms of frequency, most of the relationships, the pairwise relationships center around 0, which is actually somewhat similar to the reference line, indicating that they're actually truly independent. So there doesn't seem to be actually to be a high degree of population structure at the nuclear genome level. And the lines actually that define that a little bit typically share actually certain inversions, which explains why they actually might have a slightly higher genomic relationship there. So we were interested to see whether this is also somehow visible using the mitochondrial DNA variant catalog. And there actually we used, in collaboration actually with Matthew Robinson from the University of Lausanne, he was with Peter Vischer actually from Australia. And so he actually advises to use the statistical parsimony method and actually to look at possible population structure actually based on putative mitochondrial haplotypes in the DGRP. And indeed, actually, after basically cataloging based on 50 variants detected in at least 5 lines, we did actually see a sort of total of 12 haplotypes that emerged from this particular, based on this particular variant catalog. I have to say that this also involves some manual curation of the clusters, such that a particular variant or set of variants has to be shared by at least more than 5 DGRP lines in order to belong to a certain haplotype, and it should also not be shared by a central or another haplotype. So here you can actually see the number of variants in which basically each haplotype differs from, for example, here the central haplotype, and here the number is actually the number of lines that belong to actually each of the different haplotypes. So this was actually an interesting observation. We had a discussion. It might be criticized. To us, this was the best how we could actually address this particular problem, but I'm, of course, always open to actually suggestions how we could do this better, or whether you think this is invalid and so forth, but at least based on the advice we were able to gather, this was a sound way to actually specifically address this particular problem. So now we have a sort of haplotype population-based population structure in the DGRP, and so we can actually now move forward in the DGRP with other analyses, and one of them was actually that we were particularly interested in is to see basically whether there were certain ratio distortions in terms of mito-nuclear interactions. So are, for example, certain haplotypes basically disfavored or favored with respect to the nuclear genome, right? And so by looking at genotype ratio distortions, right, we found roughly 1,845 of them, of which 22 were complete. So for those that are not immediately familiar, what this actually means is that the allelic combination basically is biased. So there's an imbalance there. For example, in this case here, you have the nuclear reference and then the mitochondrial reference. And here you can see that if you have the mitochondrial alternative allele, then you have zero basically occurrences, or the frequency is zero, which means that basically this genotype ratio distortion is actually complete. So this, of course, defies any kind of statistic here. So it means that basically there's a severe bias against basically having this particular allelic combination in the DGRP. And so now when you actually browse through the genome, right, and you look actually where these distortions actually occur, and you actually relate that to haplotype, you can see that actually certain regions are definitely basically biased versus specific haplotypes, as is colored throughout actually the chromosomes. And one actually stood out. By far the top is sex lethal, which we were actually very puzzled with. It actually contains the highest number of genotype ratio distortions. Is specifically linked actually to haplotype 1, mitochondrial haplotype 1. Why sex lethal, we're just not sure. It has already been reported to actually be responsible for mitochondrial maintenance via dosage compensation. But of course, this remains to be verified. But it is nonetheless very interesting that among the most significant GRDs, basically, sex lethal actually was repeatedly emerging. And so this might be interesting to follow up. Nonetheless, you can clearly see that basically certain regions are basically biased for certain haplotypes, which made us conclude that large regions were particularly associated with certain GRDs for a specific mitochondrial haplotype. Hence, that mitochondrial population structure seems to be reflected on the nuclear genome. Now, one of the questions we, of course, had is, what does this mean functionally, right? And so for that, we actually forced one of the combinations, basically, via backcrossing, where we actually then forced this reference and alt-mito combination to see basically whether this kind of forced allelic combination would have specific phenotypes in terms of overall fitness. But to our surprise, no matter how we actually looked at it, we did climb assays, we did aging assays. Reviewers also suggested us to look, for example, for the same type of fitness phenotypes, but then on, for example, poor diets to see if, for example, stress mattered. We actually could not find any actually reasonable significance or difference between this kind of forced allelic combination and the sort of natural occurring one. So based, of course, on this limited actually observation of lack of phenotypes linked to these kind of forced allelic combinations, we concluded that most of these GRDs may actually reflect standing population upon establishing the DGRP rather than actually representing actual genomic incompatibility. So remains to be tested, but we were definitely puzzled actually by the lack of phenotypes when we forced this allelic combination. And this actually is something that has been observed even actually across species when you actually force a mitochondrial, one mitochondrial genome, so to speak, into even across species, for example, simulants from Madagascar, that this actually doesn't seem to actually produce a lot of very severe phenotypes, which I find actually very interesting. And then finally, of course, we were also interested to see, kind of summarizing now the overall study, and the main goal was to see whether we now could actually relate mitochondrial DNA variation to phenotypic variation. And so for that, actually, we assembled a set of 259 non-correlated phenotypes which had already been gathered for the DGRP. We considered actually 34 mitochondrial DNA variants with a minor allele frequency larger than 5% in this case, and actually then linked this to the haplotypes as well. We first actually looked simply for variation among the haplotypes and then did a pairwise Tukey test to see actually which haplotypes were then significantly different in terms of overall phenotypes. Now, when you apply this particular procedure, 12 phenotypes passed, and most of them actually were related to metabolism and stress response. which of course could make sense given that we're actually dealing here with mitochondrial DNA variation, and we of course appreciate the role of the mitochondria in these particular biological processes. One of them actually was really peculiar and was kind of interesting, and this was the following actually. It was food intake in males, which is shown here. So this is actually based on data from Garlopov et al. in a previous publication. And here you can actually see the different haplotypes, as well as the food intake for all the fly lines actually linked to this haplotype. And here we found that, for example, mitohaplotype 1 was clearly significantly different from that of mitohaplotype 5, right? So you can see a significant difference between those two, right? So does that actually mean anything? So we addressed this in several ways, but I'm going to just show you actually data from one large experiment. And this is where Rule actually devised a balancer crossing scheme to actually swap the mitochondrial genome. So the idea here is to actually do several back crossings in order to actually arrive at a mito-nuclear genome combination that is actually shown here, in which, for example, here you only take, for example, the MH5, so the mitochondrial haplotype 5, together with basically the nuclear genome of the mitohaplotype 1. And then, of course, the vice versa. Here you have mitohaplotype 1 together with the nuclear genome of mitohaplotype 5. And then, of course, you compare that to the original, basically, genotypes, which are here already listed or shown in this particular original plot. And this is actually not necessarily our data. So the hypothesis would be that, of course, if this is really true, that perhaps basically, and mitochondrial variation is really involved, that basically by simply swapping the mitochondrial genome, you would actually transform a low feeding line actually into a high feeding one, whereas basically a high feeding line, by simply swapping also its mitochondrial genome, you would actually transform into a low feeding one, right? Indicating that by simply swapping the mitochondrial genome, you would actually alter the feeding behavior actually of the respective flies, right? And so this is actually what we saw. So here basically, this is the data, and then we can look at the conclusion. So here again, we have the reproducibility of the assay, namely, if you have the MH5 haplotype with the nuclear genome, basically, you see, again, a higher food intake than the MH1, right, the red. So this is nicely reproducing what I've shown here, and this was not our data, now this is our data, right? Interestingly, when we actually swapped now the mitochondrial genome into a high-feeding nuclear background, right, then we saw actually very little change, okay? But perhaps most interesting, when you now swap basically the mitochondrial genome, the haplotype 5, into a low-feeding nuclear background, so to speak, then all of a sudden we actually saw a significant increase basically in the overall food intake of those respective flies, okay? So to us, this was actually really cool. It basically means that, again, by changing the mitochondrial haplotype, only the mitochondrial haplotype, you can turn basically a fly that is a low food intake fly into one that actually takes in much more food. So we concluded here that high food intake nuclear DNA seems to be dominant because you cannot really lower the food intake with a lower mitochondrial haplotype, lower food intake haplotype, mitochondrial haplotype, but you can actually change a low feeder actually into a high food intake one by changing here this particular haplotype. So I hope this was clear, and I thought this was actually a very nice demonstration also of why actually Drosophila genetics as well as this particular resource is so cool, because these type of experiments, of course, you could never do in humans. And so really showing actually the impact of the combination of mitochondrial DNA with nuclear DNA and how this actually might affect your food intake, okay? So the conclusions of this part, and in blue actually are things I didn't actually have time to talk about, is that we now extensively characterize mitochondrial DNA variation in the DGRP. We confirmed the existence of heteroplasmy, which is also, of course, a little bit of a worry. If you actually have a heterozygous variant, is this actually segregating in the population, or is this simply actually at the individual fly level? We actually find instances where this is really within one fly. Can't say whether it's actually in the same cell, but at least it's in the same fly. We also developed a new method to identify nuclear mitochondrial transposition, so to speak, where basically some of the mitochondrial DNA is inserted in the nuclear genome. This is a worry when you actually call mitochondrial variants, because they may actually be biased by the same stretch of DNA that is actually embedded in the nuclear genome. And so we actually devised a new strategy to detect those kind of nums, as we call them. We found actually several more that were not detected so far, but we showed that this actually does not affect the variant calling. And at least based on the identified haplotypes, using the methodology that I've discussed, we did find actually population structure, not necessarily at the nuclear genome level, but at the mitochondrial DNA level. We also showed actually that we have phenotypic variation in some of the complexes of the mitochondrial electron transfer chain. There's intergenotypic effects that actually seem to dominate, however, over haplotype effects. So that's why I didn't really mention it. So the intergenotype variation is actually larger than that of the haplotypes. This is what we found. GRDs are widespread in the fly genome, but the tested complete GRD did not affect fitness. So is this reflecting standard population structure? Is more of a question mark, given that this is, of course, an N of 1. But it seems to sort of be consistent with the literature that we were able to find. And then we showed that basically mitochondrial DNA haplotypes seem to affect mostly stress and metabolism-related phenotypes in the DGRP. And we verified one actually experimentally where we could show that actually swapping of the mitochondrial haplotype, so to speak, changes actually the food intake in males. So this has recently been published. I encourage you to go to this particular paper for more details, and of course I'd be happy to discuss it also afterwards or at any time if you have questions. I'm going to go to an unpublished story now that hopefully you will appreciate. It's again actually going on this aspect or ambition of our lab to at least try to molecularly resolve some of the observed phenotypic variation in DGRP by, for example, looking at different kinds of molecular layers and then interpreting that in terms of the observed phenotypic variation. And so the one I'm going to talk about, and I mentioned it also over coffee, is actually circadian variation, or specifically how does genetic variation impact the molecular circadian clock. So this is work actually done by a postdoc in the lab, Antonio Morelos-Filho, who actually has a history studying the circadian clock, actually initially in Alex Stark's lab, and then he came to my lab to continue this work and incorporate also this aspect of genetic variation. And again, Masha was fantastic in actually performing most of the bioinformatic analyses. So I think circadian variation is something that speaks to all of us, because I think we can clearly identify which kind of circadian phenotype we have in terms of, are you an evening person, so not a morning person, or are you a morning person and therefore not an evening person? I think there's even now a third phenotype, the nappers. I think I'm a napper, which basically means that you are doing well morning and evening, but around noon or after lunch, you need a nap in order to kind of go back to it. So there seems to be more phenotypes than we initially appreciated, but I think this idea of circadian variation is something that we probably experience on a daily basis in our lives. If we sit behind a computer, are you sitting behind your computer at 11 o'clock in the morning, or are you already there at 8? It probably kind of is reflective of your natural physiology or circadian physiology, so to speak, right? And so, again, Drosophila is a fantastic model, because I probably do not have to explain to you that actually a couple of years ago, the Nobel Prize was awarded actually to Drosophila researchers for unraveling basically how the circadian clock works in Drosophila, and this turned out to be conserved in mammals, of course, where, for example, here you have clock and cycle, which basically regulate the expression of period and timelets, which then forms this negative feedback loop where period and timelets basically inhibit clock and cycle, and as such, basically, you start to generate oscillations, okay? That there is actually natural variation among Drosophila lines, we already knew. This is actually really not new whatsoever. For example, already more than almost 20 years ago now, these were actually phenotypic readouts of circadian variation, for example, in the Cantonese and Oregon lines. And what do you see here? I have to kind of guide you through it if you're not familiar with it. Here you basically have whether the light is on or not. So this is in dark. Then you put on the light, and then you basically go back to the dark. And what you see here basically is that right before the light goes on, there seems to be activity. So this is an activity readout. Then the flies go back to sleep, right? And then basically right before the light will go out, there's again activity, right? So this is entrainment, because basically we decide when the light goes off and when the light goes on, right? But what happens if you remove this light stimulus? What then emerges basically is a natural, is the natural clock that actually is present in those specific lines. And this is shown here. So basically here we go to a dark, dark phase. So this is light, dark, and this is dark, dark, okay? And what you immediately see is this shift basically to the right, which basically means that basically the clock is getting longer and longer in those lines. So it takes them longer and longer to reactivate themselves, basically, because they no longer experience the light, so they're basically dependent on their internal clock in order to activate themselves. And actually the opposite is seen in the Oregon line, where basically they're getting shorter, the clock gets shorter and shorter, so they're basically waking up faster and faster in a certain sense before the time point where they were actually initially entrained, okay? And so one of the big mysteries, I think, in any kind of species is, where is this actually coming from, right? How is this actually encoded molecularly in the actual organism, right? So in order to try to study this, at least to a certain extent, what we try to do now is to actually look at molecular gene expression differences, basically, among all the 140. We took 140 independent fly lines, okay? And so go from genotype to phenotype through basically the detection of gene expression variation across the circadian clock, right? Now, in order to do this, you probably will agree with me that we first have to establish some kind of baseline. So what we initially did is to simply actually map gene expression, circadian gene expression in a wild type line, in this case white minus. So the idea is to do this every 2 hours for 48 hours across 4 different tissues, okay? Where the initial 12 hours is done in the light, and then in order to really look at circadian gene expression, the other time points basically are taken from flies exposed to the dark. And so if a gene actually keeps on oscillating, you know that this is due to the circadian clock and not simply because of a reaction to the light, okay? Now, we discussed it a little bit with Christian, so for that, tends to be expensive. Gene expression is an expensive assay, so a couple of years ago, and this culminated in a much larger project than we had ever thought, we kind of devised a new way of actually doing large-scale gene expression analyses, making or generating this method that we now call BRB-seq, or bulk RNA barcoding and sequencing. And it basically really is based on the advances that single-cell transcriptomics have made, in which we simply multiplex actually through the addition of a barcode very early on actually in the process all the samples together, and then everything else basically is done in one bulk tube, so to speak, where we do the reverse transcription, and then we'd also do second strand synthesis by RNA displacement. The advantage of that is that this is actually very cheap. You can do this with a high amount of samples, and so this is roughly about €5 per consumable cost per library, right? It is 3' end, so if you're interested in splicing dynamics and so forth, this method is not for you. But if you're interested in actually doing large-scale transcriptomics in a very cheap fashion, then this might actually be a method that you want to consider, okay? So using BRB or BRB-seq on these 96 samples that I mentioned just a slide before, we were now able actually using, Marsha was able using GTK cycle, which is actually well established in the field as a very good approach to detect circadian gene expression. We were able to detect 1,729 genes that actually cycle in a circadian manner in the flies. Most of those basically cycle in only one tissue, so you can clearly see these black bands here actually sort of walking or waving through the plot, right? So this clearly shows you basically high expression and low expression, and it's kind of nicely waved through, right? In each of the individual tissues, okay? And then here you actually see some that actually are in 2 tissues, then you have some that are actually present in 3 tissues, okay? And interestingly, we found actually genes, 14 of them, that were actually found cycling in all 4 tissues. Some of them you will immediately, hopefully, recognize, timeless, CRY, clock, and so forth. So these are actually already well established. Period is here as well, so you can nicely see the oscillation here. But interestingly, 7 of them actually are potentially new circadian regulators. They have never actually been really linked to the clock, but they're clearly actually cycling in all tissues, and Antonio actually knocked them down, and at least 5 out of the 7 of them actually seem to have clear circadian phenotypes, okay? So this is actually very encouraging in terms of actually getting a better understanding of the base mechanics of the clock itself. So what you can then do is to simply ask yourself, well, what is actually responsible for these oscillations? For example, first of all, on a general basis, right, in all tissues, but also very interestingly, why do certain genes basically oscillate in one tissue but not in the other? What is actually causing that, right? And so in order to study this, Masha applied this particular program here, Dynamics Gene tree, which actually infers regulatory information based on time series. I think it's a really cool kind of algorithm. And so what she is able to do is sort of generate a gene regulatory network that actually depicts all the transcription factors that seem to somehow be involved in mediating either ubiquitous circadian gene expression or a tissue-specific circadian gene expression, okay? And so if we now walk through this plot, because typically these plots are not readable, but we can still try to make sense out of it. If you look at the central nodes, then what you see is that these are actually the nodes that we would expect, because these are all actually regulators that we know have a clear central role in mediating circadian gene expression. But to us, what is actually more interesting are these kind of submodules here, in which basically the master regulators, circadian master regulators, are connected to smaller modules composed of these tissue-specific cycling transcription factors. So there seems to be a second level of hierarchy here in mediating circadian gene expression, right? Where basically the core regulators of the circadian clock cooperate basically with tissue-specific regulators, which then also actually cycle, but only in their specific tissue, right? Because 37 out of the 38 tissue-specific cycling of 37 out of the 38 of these transcription factors basically only cycled in their respective tissue, even though they were expressed in other tissues as well. So there seemed to be some kind of cooperativity between the central regulators and tissue-specific regulators, which then also cycle in their tissue, even though they can be expressed in other tissues as well. But then if they're not cycling in these other tissues, okay? Just to kind of give you a glimpse of the type of gene regulation that might underlie circadian gene expression. But now we want to go back to our original story, basically, of trying to link genotype to phenotype and actually answer this question: how much circadian gene expression variation is there actually in the DGRP, right? And so for that, basically, if you really want to answer this question, we calculated that if ideally we sample each line every 2 hours for at least 24 hours for 140 lines, and we take, for example, at least 3 tissues, the Malpighian tubes we kind of discarded because those gave us actually problems in generating actually the transcriptomes in the wild type, the white minus, you still actually get at 5,040 transcriptomes, okay? This is a lot. I don't think I have to convince you. So this was actually not feasible, was sort of disappointing. And so we said, how can we still actually try to address this question, but in a way that we can vastly reduce the number of transcriptomes that we would need in order to actually address circadian gene expression variation. And so we were actually inspired by a paper by Mirko Francesconi and Ben Lehner, in which they actually used this kind of experimental design to infer gene expression dynamics from single expression profiles, so from static expression profiles. The idea being is that you only need actually one time point, but by looking over an entire population, you can try to infer gene expression dynamics. So that got us actually thinking, and ultimately we came to the following design that we thought actually could help us to address this specific question. And so if you want to pay attention or understand the rest of the talk, this is a crucial slide. So if you are interested, please pay attention. So the idea here is basically that rather than actually doing every 2 hours for every line, what we decided to do is to basically go through the entire circadian clock for 24 hours, sampling each DGRP line only once, okay? But in a kind of sequential fashion, such that every 9 minutes, basically, we sample a different DGRP line, okay? So here, we start with a randomly chosen DGRP line. You know, here, it could be 1 or 21, it doesn't really matter, but let's say 1. And then 9 minutes later, we actually do DGRP2, then DGRP3, 4, 5, and so forth. So all 100, I think it's in total actually it should be 160 indeed. You know, I had 140. We actually ultimately sampled 160 lines. We actually went through the entire circadian clock by simply sampling actually all of the lines throughout this 24 hours, right? So the idea being that actually if they're completely consistent with what we would expect, they would actually nicely together reconstitute basically the overall oscillation that we would expect to see if you would have actually sampled only one line but every 2 hours throughout the entire clock, right? I don't know if this makes sense, but let's try again. So this is the timeline of timeless expression in the gut, for example, in the wild type or the white minus, right? So you can nicely see this oscillation. So the question is whether you can actually reconstitute this same expression profile, but simply by actually adding dots here where each dot actually would be derived from a different DGRP line, right? It simply actually shifts from the dots would simply be not only different in time, but also in DGRP line. That's the only caveat here. So if you understand this, then this is great because then you will understand the rest. And so here it is. So each dot on this particular plot is a DGRP line. And what you can see is that overall, most of the DGRP lines actually nicely follow the pattern that you would expect to see, right? Indicating that actually this particular DGRP line, for example, this one, is exactly actually showing, for example, expression of timeless at exactly the time point where you expect it to be, right? But of course, the interesting part, and this is true, of course, in the gut and the fat body, but the interesting part is, what are these orange dots, okay? Because the orange dots basically defy the time point in a sense that the gene expression of timeless is not where it's supposed to be, right? For example, here in this case, this guy here should be up there, but it's not. So does that mean that basically this guy is actually 6 hours behind or it's 7 hours ahead, right? This is actually what it comes down to, okay? And so now, this is actually really interesting because it turns out that this is starting to be applied on human samples more and more because it's, if you actually sample a human, right, from a specific tissue, it's actually interesting to know at which circadian time you sampled this particular human. But you only have one time point because you didn't sample the liver of this human multiple times throughout the circadian clock, obviously, right? And so researchers in the field have started actually to think of how to solve this, and one of the ways that was proposed is this molecular timetable approach, where you actually use a series of time-indicating genes. These are typically, but they don't have to be, core clock genes. And together, basically, you do sort of machine learning, actually, on your data to kind of try to infer, based on these time-indicating genes, what the actual time is of the circadian time of your particular line, based on, basically, this set of indicative genes, right? So you basically then infer the time from this line, and you actually compare it to the time at which you sampled the line. And if that time point basically is the same, that's good, right? Because that benchmarks your approach. But if it's different, it basically means that there is circadian gene expression variation, right? And so if you do this, and now I have to just guide you through the plot. So here you have 3 different tissues. Here you have many DGRP lines with their respective numbers. And here you basically see circles that keep on getting bigger, and they keep on getting bigger because they actually link to the hour or the number of hours where basically they deviate, where the expected time is actually deviating from the sampled time, okay? And so here, for example, you have a line in which basically all of the gene expression actually in all 3 tissues basically is significantly basically deviating from what you would have expected. So this clock basically is definitely disturbed, okay? There's also one here like this, for example, where basically all the gene expression profiles that we actually got from this line on the 3 different tissues actually were not where they're supposed to be. They were significantly either lagging or getting ahead. Here in this case, almost 10 hours, right? And so what is interesting is what you also hopefully appreciate is that in some, for example, like in this guy, let me take a good example. In this case, guy, for example, let's see, and this guy only has actually molecular variation, gene expression variation in the gut, and in the fat body and brain, it's actually very small. It centers actually around basically it's not really different from what you would expect in terms of overall noise level. Here you have, for example, 2 tissues where you have actually significant gene expression variation. Here again, you have only, in this line, you also only have really gene expression variation significant in the brain, and so forth, right? So you have some lines where you have gene expression variation, circadian gene expression variation in all 3 tissues. In some, it's only in 1 tissue. And to me, this is absolutely remarkable. So that means basically that there's extensive molecular or circadian molecular variation, but mostly in a single tissue, which basically means that genetic determinants act mostly at a tissue-specific level. We can discuss afterwards how this could be. Maybe these are tissue-specific regulators that act through variants that only act basically at a tissue level. But it also means that flies actually exhibit circadian level expression organismal desynchrony. It means that basically your organs normally have to be synchronized, but in some flies, basically, the gut, for example, is lagging the other tissues because there's sort of variation that actually imposes a lag or a gene expression change as compared to what you would expect to see in a wild type. So to me, this is astonishing that there's actually this kind of desynchrony between tissues, where in one tissue, basically, you would have a gene expression, circadian gene expression variation, but in the others, there's not. So how this is actually handled physiologically is, of course, very interesting to think about, right? Okay, let's see. I'm gonna. Yeah, sure. Right. So basically what you can do is you can benchmark your approach. So what I'm skipping here is that we basically did this, we still actually mapped 4 DGRP lines where we actually did the entire circadian time. And then we actually benchmarked our approach and we saw actually what is the noise level, right? How good is our method actually predicting the actual time from the sample time, right? And basically everything that then is beyond the noise level is what we actually considered significant, right? So that's why basically here you have some small circles, but this is maybe 2 hours difference, but we consider this within the margin of error, so to speak, right? So anything that is way beyond this, we consider actually that we can call this. So this is still quite conservative, right? I mean, it depends actually where you put the threshold, but we used a very conservative threshold such that we are certain that this kind of gene expression variation here is truly actually present in that particular line. Does that answer your question? It's a good point. It's a very good point. Well, I mean, like I said, I mean, it's always difficult to say. This is actually one of the most difficult parts. Basically, where are you? Are you 7 hours ahead or 6 hours behind? So there is actually a way to do that. We don't really make these calls here. Basically, we just say, okay, there's an overall time difference, so to speak, of, for example, an X amount of hours. In what the paper we're now preparing, we actually discuss how you actually could further resolve this, and there's different ways how you could go about it. So ultimately, this could be possible, yes. But I mean, for the overall concept, it doesn't really matter whether you're ahead or because it's a sort of semantic in a way. But it probably might explain, it's important. Don't get me wrong, but whether it's now the most important thing that we try to say is that there's clear variation, right? Because how to interpret then, ultimately we would have to then do circadian behavior tests basically on all these different lines in order then to couple the molecular readouts actually to the actual behavioral readouts, right? Which is something I could discuss afterwards as well. Is something that in a way has been done, and I'll get to it in a moment actually. Okay. So now we actually try to kind of get a mechanistic understanding as to why this variation actually occurred and what the impact could be on a phenotype, right? And so we started to actually initially focus on those where there's actually tremendous variation, gene expression variation, and circadian gene expression variation in all 3 tissues, okay? And so, of course, we did a mutational scan to look for example, mutations in the core circadian genes. And we didn't really see something that was very obvious or that was actually generating a null mutation, right? So we were kind of at loss to actually try to explain this, so we went back to the phenotype of this particular line. And here again, you have these same behavior plots, where again, you do the dark lights, you look for activity before you put on the light, and then you kind of see this activity increase, and then it tapers down, goes up again before the dark, and so forth, right? So this is this line, basically, where we see this tremendous variation in all 3 tissues. And overall, you can actually see that there's not a big difference. I mean, it's a bit more smoothened, right? But overall, it doesn't seem to actually really be significantly different, okay? But then Antonio did actually a very nice trick. What he actually did is to simply apply a light pulse for 20 minutes, which actually is known to induce actually a phase shift in wild-type lines. And this is nicely illustrated here. So if you put the light on here, this little white rectangle here, you can see that the flies now all of a sudden adapt themselves to this light pulse, even though it was only 20 minutes, right? So they get re-entrained actually because of this light stimulus, okay? But hopefully you will appreciate that even though this same light stimulus was applied, these guys were actually not responding. They still are actually entrained exactly the way they were actually before we changed actually the light pattern to these flies. What does that mean, right? It means that somehow basically there's a defective light response and that there's light-independent entrainment. This is also very puzzling in the field. How this is actually particularly done is also not entirely clear, but at least the data tells us that there is a defective light response, okay? Now, in order to actually look at where the mutation might be that causes this, if there is actually one single mutation, we did chromosome swapping. So Antonio did chromosome swapping, and again did the same thing. So he added this light pulse, swapped chromosomes, and then just simply looked for at which situation when you swap each chromosome don't see this phase shift that you normally see in the wild type, right? So what he discovered basically is that this was only actually occurring when he swapped chromosome 3, indicating that somehow this rescued basically the mutation, or in this case basically it tells us that the chromosome 3, when you actually put it in a wild type fly, or you swap the chromosome of DGRP 796 in a wild type line, that actually, again, causes this line to no longer respond to light, okay? This is what it basically says, right? And then we were puzzled because one of the clear genes that is on chromosome 3 that is responsible for light response is CRY, one of the core circadian clock regulators. So why didn't we actually originally see that there might be a mutation there? And the reason is the following. So we went back to that gene, and what we discovered is that indeed this gene actually has a small mutation. It's a deletion of 6 base pairs. So this is actually in frame, so we never actually tagged it as a serious mutation because, you know, as I already said initially in my talk, the DGRP is highly polymorphic, okay? It's also not in sites that we know are actually crucial because these are some residues basically that are known to be functional within the CRY protein. But we nonetheless then try to kind of figure out whether this impact or this variation actually here has any impact whatsoever, right? And for that actually we went we engaged in a very nice collaboration with Matteo Del Perraro's lab at the EPFL, where we actually looked at the structure of this CRY protein, okay? So this is the Drosophila one, the wild type, right? And now we overlaid it actually with the one from the DGRP 796. So this is of course simulated. We didn't actually crystallize it, but this is simulated. And as you can see, there is actually no real change. So this was sort of disappointing, okay? So it might not actually lead to anything. But then actually we looked at a couple of clear and key residues, which are these tryptophanes basically here, which are responsible or thought to be responsible for actually the electron cascades in responding to the light. So electrons are basically channeled from one tryptophane to the other and ultimately basically activate the light response pathway, right? And then very interestingly, lo and behold, as we say, right, if you now actually look at the tryptophan in the Drosophila in 796, what you see is that this small deletion that is in frame nonetheless actually flips basically one of the tryptophan on the other side of the helix, right? Basically indicating that the electron transfer here is actually simply disrupted. So the initial one basically no longer can accept the electron. It can, but it will never actually transfer it to the other tryptophans because it's on the other side of the helix. So this is not a null mutation whatsoever, but it actually simply kills the ability of this protein actually to probably transfer the electron transfer chain, the electrons, and then induce the light stimulus. So we've done all kinds of functional tests on top of that to prove that indeed this CRY is indeed responsible for the observed phenotype, nicely showing in a way that how we went from observing circadian molecular variation in this line in all tissues to understanding mechanistically why this is in finding the causal mutation through this specific mechanism, right? So with that, I no longer will bother you with more science. I will simply actually summarize what I just talked about in the second part. I think we have now a very rich data set for circadian biology with high resolution and more than 700 samples or transcriptomes. We found putative new clock genes. We found pervasive, mostly tissue-specific circadian gene expression variation. And this variation, we think, is mediated by a multi-layered circadian gene regulatory network involving central regulators that corroborate with local regulators that cycle in a tissue-specific fashion. And genetic variants may act through these local regulators to affect circadian expression, or may be non-intuitive but highly penetrant, as the one that I actually showed you at the end. So with that, and probably going a little bit late here, so thank you very much for your attention. I want to again thank all the people involved, specifically Masha, who was responsible for most of the bioinformatic analysis for the 2 parts. I want to thank Antonio and Rul, actually, who are now alumni of the lab, for their fantastic work. These are our collaborators. These are the funding sources. This is the EPFL, if you're interested. And I'm very happy to answer any questions you might have.